Welcome to the Deep Dive, where we take a stack of sources and, well, dive deep to pull out the most important knowledge for you.
Think of it as getting the essential insights, the surprising facts, and just enough context to feel truly well informed without having to spend hours poring over the details yourself. We're here to guide you through the fascinating stuff.
And today, we're looking at a source from a name you might find surprising in this context. We're doing a deep dive into a biology textbook written by none other than H.G. Wells.
H.G. Wells, really?
The visionary behind War of the Worlds and The Time Machine.
That's right. Before he imagined alien invasions and journeys through time, Wells was actually a trained biologist. The source material for our deep dive today is his textbook of biology, Part 1, Vertebrata, first published back in the late 19th century.
And this wasn't exactly light reading, was it? It was written specifically for students getting ready for examinations using a very particular teaching method they called the type system.
Yeah, the type system. The core idea, really, was to study just a few representative animals in meticulous detail, almost like blueprints, you could say, to understand the fundamental general principles of biology that apply much more broadly.
So focusing in to understand the big picture.
Exactly. It was designed to teach students how to observe precisely, how to deduce, how to verify things, which was especially important for those studying alone, as the introduction by G.B. Howe's notes.
Right, because this was also used for correspondence courses, wasn't it? So clear instructions were vital.
Absolutely. So the point wasn't just rote memorization or pure cram, as they rather bluntly put it, but truly grasping the structure and function by looking closely at real examples.
Okay. And our mission today?
Well, our mission is to extract the most interesting and important insights from this pretty detailed text by comparing these different animals through Wells' eyes. What did he think was key for students to understand about life over 100 years ago?
To do this, the book focuses on four main animal types that sort of stand in for the diversity of vertebrates. You've got the rabbit,
Representing mammals,
the frog,
for amphibians,
the dogfish,
representing fish, cartilaginous fish specifically.
and then this creature called Amphioxus.
Yeah, Amphioxus, a simpler lancelet-like creature.
Yeah.
It's often considered a proto-vertebrate, kind of like a basic sketch of what early vertebrates might have looked like.
So by comparing these four, you get a really clear picture of the major steps and variations in the vertebrate story.
That's the idea, yeah.
So let's start where the book does with the philosophy behind it and the importance of, well, getting your hands dirty, so to speak. Wells and Howes really stress the value of this type system and practical work.
Oh, definitely. They're quite adamant that while learning from a teacher with actual specimens is obviously best, if you're learning alone, like many of these students were, then incredibly precise instructions are absolutely essential.
And the type system provides that structure.
It provides that discipline needed for detailed observation, for manipulation dissection, basically, and for deduction and verification. It's rigorous.
They make a strong point that simply reading about things just isn't enough. You have to do the practical work, the observation, the dissection to truly confirm and complete your knowledge.
Exactly. The book even includes a whole syllabus just for the hands-on part. It forces you to see the reality behind the diagrams.
Okay, let's jump into the heart of the book then, comparative anatomy. The text goes deep on each animal individually, but the real power, as Wells shows, comes from putting them side by side and seeing the differences and similarities. Let's start with something fundamental, the digestive system.
Absolutely. And right away, just comparing the rabbit and the dogfish tells you a lot about how diet shapes anatomy. The rabbit, being an herbivore, has this really long, coiled gut. You have to picture it as a complex processing plant, really, designed to break down tough plant matter.
Right. It starts with the esophagus, leads to the stomach, then a very long small intestine and a large intestine. And it's all sort of neatly held in place by these membranes, the peritoneum and mesentery.
Yeah, and the text makes a point about herbivores needing to chew, thoroughly masticate, that's the term, to mix food with saliva, which starts breaking down starch right away with an enzyme called ptyalin.
Which is different from carnivores.
Right. They contrast this with meat eaters like cats or dogs, where chewing is more about ripping and tearing than grinding. Their teeth are different, too sharp for gnawing. Rabbits don't even have those pointed canine teeth.
And inside the rabbit.
Well, inside the rabbit's digestive juices, like pepsin in the stomach for protein and various enzymes from the pancreas, work on proteins, fats, and carbs. Bile from the liver helps with fat digestion, too.
And the rabbit has that really distinctive feature, that huge pouch called a cacum and a little tail off it, the vermiform appendix, which we still have, sort of.
Yes. And they point out this massive cecum is drastically reduced or even missing in carnivores. That strongly suggests it's crucial for processing cellulose and other plant fibers that herbivores eat. It's where a lot of microbial fermentation happens.
Okay, now contrast that with the dogfish, a carnivore.
Much simpler. Its gut is shorter, straighter, but to make up for the lack of length, the intestine has this clever structure inside called a spiral valve.
A spiral valve.
Yeah, it's like a corkscrew running down the inside. It massively increases the surface area for absorbing nutrients within a pretty compact space. Smart design. It also has a special rectal gland near the end, which helps it get rid of excess salt from seawater.
And the frog, where does it fit in? Kind of in the middle.
The frog's gut is, a bit less specialized than either. The stomach isn't as clearly distinct from the esophagus. The intestine has a U shape, but none of the rabbit's sort of saculations or that huge cecum.
And it has that cloaca thing.
Right? A key difference we see here, and in the dogfish too, actually, is that the end of the gut opens into a cloaca. That's a single chamber that also receives products from the urinary and reproductive systems. Totally different from mammals like the rabbit, which have separate openings for everything.
And finally, the simplest one, Amphioxus.
Its gut is the most basic imaginable, just a simple straight tube from mouth to anus. And the anus, weirdly, opens on the left side of the body.
The left side, huh?
Yeah. It filters tiny food particles from water passing through its pharynx. There's no complex stomach or coiled intestines, just a simple pouch that acts somewhat like a liver. It really shows you the kind of starting point for the vertebrate digestive system.
Okay, moving from processing food to circulating what the body needs, the circulatory system. We're talking blood, vessels, heart.
Right. And blood itself has different components: plasma, white cells, and red cells that carry oxygen. And the text notes an interesting detail. Mammal red blood cells are these non-nucleated discs, unlike the nucleated ones you find in the other types, like frogs and fish.
Different efficiency, maybe.
Possibly, yeah. Now, the rabbit, as a mammal, has what's called a double circulation. You can imagine two separate loops running at the same time.
Okay, two loops.
One loop pumps blood just to the lungs to pick up oxygen, and then back to the heart. The other loop takes that freshly oxygenated blood and pumps it out to the rest of the body, then back to the heart again.
And the heart reflects this double system.
Exactly. It's got 4 chambers. You could think of it as 2 pumps side by side. This setup keeps the oxygenated blood completely separate from the deoxygenated blood. Very efficient.
And one side is stronger.
Yes, the left side, the left ventricle specifically, which pumps blood to the entire body, has a much thicker, more muscular wall than the right side, which only has to pump blood to the nearby lungs. And there are valves, of course, to make sure blood only flows in one direction.
There's more to it than just the heart main vessels, though, right? Portal systems, lymphatics.
Yeah, it's complex. You have intricate networks of veins bringing blood back. And these portal systems are interesting. That's where blood flows from one capillary bed, say in the intestines, directly to another capillary bed, like in the liver, before it goes back to the main circulation. The liver portal system is key for processing absorbed nutrients.
And the lymphatic system.
That's a whole separate network of vessels that collects fluid from tissues and plays a big role in immunity. It eventually drains back into the main blood circulation. And of course, breathing air into lungs allows for very efficient gas exchange in tiny air sacs, the alveoli, right next to the lung capillaries.
Okay, so that's the complex mammalian system. Contrast that with a dogfish.
Totally different. This is a single circulation system, which is typical of fish. The heart, which is kind of Z-shaped with just 2 main chambers plus some collecting and exit areas. It pumps only deoxygenated blood.
Only deoxygenated. So where does it get oxygen?
It pumps that blood straight forward to the gills. The gills are where the blood picks up oxygen directly from the water flowing over them. From the gills, the now oxygenated blood flows directly out to the rest of the body, delivers the oxygen, picks up CO2, and then loops back to the heart, deoxygenated again.
So heart, gills, body, heart, one loop.
Exactly. It's a simpler, maybe less powerful system, but it's perfectly suited for aquatic life, where oxygen is absorbed differently.
And the frog. We keep saying it's intermediate.
It really is here, too.
Sure.
The frog's heart is interesting. It has 2 receiving chambers, the auricles, like the rabbit. One gets oxygenated blood from the lungs and skin.
Skin. Frogs breathe through their skin.
You do. Significant gas exchange happens through their moist skin. So oxygenated blood comes from lungs and skin to the left auricle. Deoxygenated blood from the body comes to the right auricle, but they only have a single pumping chamber, the ventricle.
Ah, so the blood mixes in there.
There's some mixing, yes. Oxygenated and deoxygenated blood aren't kept completely separate before being pumped out. It has arteries going to the lungs and skin, and others going to the body. It's less efficient than the mammal system, but better than the fish system for an animal that lives both in water and on land.
And it still has those portal systems.
Yep, it retains portal systems similar to the dogfish, including one going to the kidneys, a renal portal system, and one to the liver. They even have these little pulsing structures called lymph hearts to help move the lymphatic fluid around. Quite different.
Okay, and Amphioxus. Back to basics again.
Absolutely rudimentary. There's no distinct muscular heart pumping blood forcefully. Instead, blood is moved along by the slow contraction of certain major vessels, particularly one running along the underside of the pharynx, sometimes called the cardiac aorta.
So no real pump.
Not like we think of a heart. Blood goes up through the gill bars.
Ah.
It doesn't even have red blood cells, mind you. It's just plasma-carrying dissolved oxygen gets oxygenated, flows through the body, and a simple liver-like structure, and then back to that main ventral vessel. It's like a faint echo of a vertebrate system, maybe showing how it started.
All right, let's talk about what holds it all together. The skeleton. Made of bone or cartilage, you said.
Yeah.
The rabbit, like us, has a bony skeleton.
A very complex framework, yes. Think of the spine. Individual bones, the vertebrae, all stacked up. Each vertebra has a main body, arches protecting the spinal cord, and various bumps and processes for muscles to attach and for connecting to ribs.
And the ribs connect to the vertebrae.
They articulate, yeah. And the skull is just a marvel of engineering a bony case protecting the brain, plus all the bones forming the face and jaws. Wells goes into detail, naming all the different bones, the basic occipitals, sphenoids, parietals, frontals, maxillae, mandible. It's intricate.
Now, the dogfish, you said totally different.
Entirely cartilaginous. No true bone anywhere in its skeleton. Its vertebrae are much simpler, like rings that are hollowed out front and back. And that flexible rod, the notochord, runs right through the middle of them, along the whole length of the spine.
Still has a notochord as an adult.
Very prominently, yes. The skull is also just cartilage, the brain case, capsules around the sense organs, the jaws, the arches supporting the gills. It's a much lighter, more flexible skeleton compared to bone. Good for moving in water.
And the frog. It starts as cartilage but ends up with bone.
As a tadpole, yes, it's mostly cartilage. But as it becomes an adult frog, much of that skeleton turns into bone. But its bones are different from the rabbit's in some key ways. Its vertebrae, for example, are hollow in the front, not flat like a mammal's.
And it has that weird tailbone thing.
The urostah, yeah. A unique long rod of bone at the very end of its spine. And another big difference. Unlike the rabbit, the adult frog has no ribs at all.
No ribs.
Nope. Its skull development is also complex, involving both cartilage turning into bone and some bones forming directly from membranes in the skin, like parts of our skull do. And its shoulder girdle, the bone supporting the front legs, is actually more complete and robust than in a mammal.
You mentioned that notochord again. Its role seems to change across these types.
It really does. That flexible rod is a fundamental chordate feature present in the embryos of all these animals. In the dogfish, as we said, it stays prominent right through the adult cartilaginous vertebrae. In the adult frog, it's reduced, but remnants persist within the bony vertebrae. In the adult rabbit, it's almost entirely gone, just tiny remnants might remain between vertebrae, but it was definitely there in the embryo, guiding spinal development. Seeing how it persists or gets replaced is a really key indicator of evolutionary relationships and development.
Okay, let's move to the systems that control everything. Muscle and nerve. The text describes 3 types of muscle tissue.
That's right. There's unstriated or smooth muscle. Simple cells, contract slowly, found in the walls of organs like the gut and blood vessels. Then there's striated muscle, which has visible stripes or striations under a microscope. These are complex fibers that contract quickly and sharply, and they're typically used for voluntary movement, like moving your arms and legs.
And the third type?
Cardiac muscle, the heart muscle. It's kind of intermediate. It's striated like skeletal muscle, but the fibers are branched and interconnected, and it contracts rhythmically on its own. The text makes a good point, actually, that calling muscles purely voluntary or involuntary isn't perfectly accurate. Your heart muscle is striated, like voluntary muscle, but you certainly can't will it to stop beating. And your limb muscles can perform involuntary reflexes.
Good point. Okay, and the nervous system, the body's communication network and, well, control center.
Right. Made up fundamentally of nerve fibers, which are like the wires conducting signals, nerve cells, often called ganglion cells, which process information. They're the officials, as Wells puts it, and then supporting tissue called neuroglia. The nervous system governs pretty much everything, from simple automatic actions to complex thought.
The simplest level of control is reflex action, you said.
Yes, that's a direct automatic response to some kind of stimulus, like, your pupil shrinking in bright light or jerking your hand away from something hot. The signal goes in through a sensory nerve, and a response comes straight back out through a motor nerve. It seems almost mechanical.
Then there's what the book calls mental action.
Yeah, that seems to involve something more than just a direct stimulus response loop. It brings in sensation, maybe basic awareness. The example of pain shunting suggests a slightly more complex redirection of signals.
And then voluntary action is the top level.
That's where stored impressions come in: memory, comparison, thought, conscious will. The text gives this nice example of a frog deciding whether or not to snap at an insect based on whether it's had bad experiences with similar-looking things before. So it's not just reflex. There's some kind of processing based on past experience.
But even voluntary actions can become automatic.
Interestingly, yes. They note that actions learned voluntarily, like walking or writing or playing an instrument, can become so practiced that they become almost automatic, quasi-reflexive. They slip out of the sphere of mind, as they put it, into habit, which lets your conscious mind focus on other things, sometimes leading to absent-mindedness.
Now, in the rabbit, as a mammal, you see a pretty complex brain with different parts specialized for different jobs.
Oh, definitely. The forebrain is dominated by these large cerebral hemispheres, which are associated with higher functions: mind, memory, complex thought, learning. Then you have other parts like the cerebellum, tucked away at the back, which is crucial for coordinating movement and balance. And the medulla oblongata at the base of the brain controls vital automatic functions like breathing, heart rate, even things like yawning and coughing.
And mammals have that special connecting structure.
The corpus callosum, yes, a large bundle of nerve fibers connecting the left and right cerebral hemispheres. This is really distinctive of mammals and allows for very efficient communication between the two sides of the brain.
Below the brain, the spinal cord runs down the back. What's its main job?
It's the main pathway for signals between the brain and the rest of the body, but it also handles a lot of reflex actions independently. It has distinct pathways for sensory information coming in, dorsal roots, and motor commands going out, ventral roots.
And there's another part of the system, too, the sympathetic chain.
Yes, the sympathetic nervous system. It's a chain of ganglia running alongside the spine, controlling many involuntary functions, fight or flight responses, gut activity, heart rate, things you don't consciously manage. And then connecting directly to the brain, you have the 12 pairs of cranial nerves, controlling senses like smell and sight, eye movements, facial muscles, hearing, swallowing, and innervating organs in the chest and abdomen.
Okay, how does the frog's brain compare? Simpler.
Generally simpler, yes, and the proportions are different. Its optic lobes, the part associated with vision, are proportionally much larger than in a mammal. The text suggests the cerebral hemispheres handle maybe lower functions compared to mammals. The cerebellum, for coordination, is much smaller, reflecting the frog's simpler range of movements compared to, say, a rabbit.
And Amphioxus, we're expecting basic again.
Dramatically different. Its central nervous system is basically just a simple tube running along its dorsal side. It's not really differentiated into distinct complex brain regions with fluid-filled cavities like the other vertebrates. There's just a very slight swelling at the front end, but you wouldn't call it a brain in the way we recognize one.
No complex regions.
No. The nerves just branch off fairly simply along its length. And for senses, it just has a basic light-sensing pigment spot near the front. Not a complex image-forming eye at all. It's a very fundamental nervous system structure.
Speaking of sensory organs, let's look more closely at the eye and the ear. The rabbit's eye, you said, is complex, like a camera.
It really is. It has these distinct layers. An outer protective coat, the tough white sclera at the back, becoming the clear cornea at the front. Then a middle coat, the choroid, which is rich in blood vessels and pigment, and forms the iris at the front. That's the colored part that controls the size of the pupil, adjusting light levels reflexively. And then the inner layer, the retina.
The retina is the light-sensitive part, right, where the actual seeing happens.
Exactly. It contains the photoreceptor cells, the rods and cones, that detect light. And here's that really surprising fact the text highlights. In all vertebrates, these light-receiving cells, the rods and cones, actually face away from the incoming light.
Wait, they face backwards.
Yes, the light has to pass through several layers of nerve cells and fibers within the retina before it hits the light-sensitive part of the rods and cones. It seems counterintuitive, but that's how vertebrate eyes are built. It's different from how many invertebrate eyes, like an insect's compound eye, are structured.
That That is odd. And there's a blind spot, too.
Yep, where the optic nerve fibers bundle together and exit the eye to go to the brain, there are no rods or cones in that specific spot, so you can't see anything that falls precisely on that point. The book even describes a simple test you can do to find your own blind spot.
Okay, what about the ear? The rabbit's ear has 3 parts.
That's the typical mammalian setup. You have the external ear, the visible part, the pinna for collecting sound, and the ear canal. Then the middle ear, which starts with the eardrum, tympanic membrane. Vibrations hit the eardrum and it passes them along through 3 tiny interconnected bones called ossicles, the malleus, conegus, and stapes. These bones amplify the vibrations and transmit them to the inner ear. The middle ear also connects to the back of the throat via the eustachian tube, which helps equalize pressure.
And the inner ear is where hearing actually happens, and balance.
Right. The inner ear is a complex structure called a labyrinth, embedded within the skull bone. It contains fluid-filled canals and sacs. 3 semicircular canals, arranged at right angles to each other, are crucial for detecting head movements and maintaining balance. And then, specifically for hearing in mammals, there's a spiral-shaped structure called the cochlea. It's filled with fluid and lined with sensitive hair cells that detect the vibrations transmitted from the middle ear bones and convert them into nerve signals. This coiled cochlea is really distinctive of mammals.
Okay, now compare that complex ear to the dogfish.
Much, much simpler. The dogfish basically only has the inner ear, the labyrinth, with the semicircular canals for balance. It completely lacks an external ear, an eardrum, or those middle ear ossicles.
Why no middle ear?
Because it lives underwater. Sound waves travel very effectively through water and directly through the fish's tissues and skull to stimulate the inner ear fluids. It doesn't need the apparatus that land animals evolved to collect and amplify faint vibrations traveling through air.
Makes sense. And the frog's ear? Our intermediate example again?
Exactly. It's kind of in between. It has an internal ear, the labyrinth with semicircular canals, similar to the rabbits, for balance. And it does have a middle ear. There's an eardrum, often visible as a circular patch on the side of the head behind the eye. But its middle ear is simpler than a mammal's. It only has a single ossicle bone, called the columella, or stapes, transmitting vibrations from the eardrum to the inner ear, not 3.
Only 1 bone instead of 3.
Right. And interestingly, the text points out that this middle ear cavity and the eustachian tube in the frog developed from what was ancestrally the first gill slit, or spiracle, found in fish. It's a great example of evolutionary repurposing a breathing structure becoming part of a hearing apparatus.
Okay, finally, let's look at the systems for filtering waste and for reproduction, the renal and reproductive organs. The book presents a kind of generalized vertebrate plan first, doesn't it?
Yes, it does, which is really helpful. It shows how vertebrates, either in their embryonic development or sometimes in simpler adult forms, actually develop a series of potential kidney structures along the body axis, appearing sequentially from front to back. These are called the pronephros, forekidney, mesonephros, mid-kidney, and metanephros, hind-kidney.
And different ducts are involved?
Right. There are typically 2 main ducts associated with these developing kidneys. One, sometimes called the pronephric duct, or later the Mullerian duct, often becomes the oviduct, the tube carrying eggs, in females, and tends to disappear or become just a tiny remnant in males. The other, the mesonephric, or Wolffian duct, often becomes the main duct carrying sperm, the vas deferens, in males. While its fate in females varies, it might carry urine or largely disappear depending on the group.
So how does this play out in our specific examples? Let's start with the rabbit.
In the rabbit, like all mammals, the functional adult kidney is the metanephros, the hindmost one. Its specific duct is the ureter, which carries urine down to the bladder.
Okay, and for reproduction?
Female rabbits have ovaries that produce eggs, which travel down tubes called oviducts into the uterus, which in rabbits is often Y-shaped or has 2 horns, and then there's a vagina leading to the outside. Male rabbits have tests that produce sperm. The sperm matures in a structure called the epididymis, which is actually derived from part of that embryonic mesonephros. From there, they travel through a duct called the vas deferens, which is the old mesonephric duct, join the urethra, which also carries urine from the bladder, and exit through the penis.
And there's that remnant structure in males?
Yes. The text points out a small, non-functional structure called the uterus masculinus in male mammals, which is thought to be a remnant of the Mullerian duct system that forms the uterus in females. Another echo of shear development.
Okay, now the frog system, how does it compare? Pretty different.
Quite different, yes. In the frog, the functional adult kidney seems to be formed from a combination of the mesonephros and maybe parts of the metanephros that are sort of fused together. And its main duct, the Wolffian duct, is less specialized than in the rabbit. It carries urine away from the kidney, but in males, it also carries sperm.
So one tube for both, and it goes to the cloaca.
Exactly. This combined urogenital duct opens into that common chamber, the cloaca that we mentioned earlier with the digestive system. Female frogs have ovaries, and their oviducts, which are the Mullerian ducts, are completely separate structures from the kidneys. They also open independently into the cloaca, often with wide openings to receive the eggs.
And how do the testes connect in the male frog?
The male frog's testes are connected to the kidney itself by tiny tubes called vasa efferentia. So the sperm actually pass into the kidney tubules briefly before entering that main Wolffian duct to travel down to the cloaca.
That's complex. And what about the bladder?
The frog bladder is peculiar. It's a thin-walled sac, often two-lobed, opening off the ventral side of the cloaca. But here's the weird thing. The kidney ducts, the ureter's Wolffian ducts, do not empty directly into it like in a mammal. Urine flows into the cloaca first and then backs up into the bladder for storage.
That is odd. Okay, what about the dogfish?
The dogfish also uses the mesonephros as its main functional kidney. Its duct system is maybe a step towards the mammal condition, as the urinary part, the ureter, is largely split off from the reproductive part, the Wolffian duct, although they might join near the end.
And it all goes into the cloaca, too.
Yes, like the frog, everything opens into a cloaca. Female dogfish usually have only one functional ovary. The right one typically develops fully. The eggs pass into oviducts, Mullerian ducts, which often fuse together near the cloaca. A special gland in the oviduct wall secretes that tough, leathery, horny egg case around the fertilized eggs before they're laid.
And the male dogfish?
Male dogfish have testes, often partially fused. Like the frog, tiny tubes, vasa efferentia, connect the testes to a modified part of the kidney, epididymis. The sperm then travel down the Wolffian duct, vas deferens, which runs to a sort of urogenital sinus or papilla, into which the ureters carrying urine also typically open. And this combined opening leads into the cloaca.
Wow. It really is quite something to compare these and see how the same basic parts, mesonephros, metanephros, Mullerian duct, Wolffian duct, are used and modified in different ways across these groups.
Absolutely. The text even suggests you can sort of mentally picture shifting the rabbit structures around, moving things posteriorly and ventrally to see how they relate to the basic arrangement found in the dogfish near the cloaca. It highlights that underlying shared blueprint.
So we've seen all these fascinating differences and similarities in the adult anatomy across these different types. This leads us naturally to another really powerful line of evidence that Wells emphasizes in the book. Development, the story of how an animal actually grows from a fertilized egg.
Right. Wells shows very clearly how studying the process of development, embryology, reveals even more striking similarities between these vertebrate types, especially in their early stages. Even animals that look incredibly different as adults often go through remarkably similar phases as embryos. It's like they're following a common developmental script early on.
It all starts with the fertilized egg dividing, doesn't it?
Yes, it starts with cell division, called segmentation or cleavage. The egg divides repeatedly, forming a ball of cells, which then typically becomes a hollow sphere called a blastosphere. And then comes a really crucial stage called gastrulation.
Gastrulation, that's where the main body layers form.
Exactly. During gastrulation, the basic body plan is laid down. The cells rearrange themselves to form distinct layers, usually an outer layer called the epiblast, which will form skin and nerves, an inner layer called the hypoblast, forming the gut lining, and often a middle layer, the mesoblast, forming muscle, bone, blood, etc. At the same time, the primitive gut cavity, the archenteron, starts to form.
Does this happen the same way in all of them?
The way gastrulation happens can look different, especially depending on how much yolk is present in the egg. In Amphioxus, which has very little yolk, it's a relatively simple process where one side of the hollow blastosphere just pushes inwards, like indenting a soft ball, forming that inner layer in gut cavity.
Simple invagination.
Pretty much. And the notochord, that primitive backbone rod, pinches off from the roof of this early gut. The nervous system starts as a flat plate on the back that then folds inwards to form the neural tube, which becomes the spinal cord and brain.
Okay, what about the frog? It has more yolk.
The frog's development is modified by the yolk, which makes the cells divide unevenly and slows down the process at the yolky bottom pole. So gastrulation involves cells rolling inwards over a lip, the blastopore lip. But fundamentally, you still see the formation of those same three germ layers, the notochord forming from the roof of the archenteron, and the neural tube forming from folds on the back, following a broadly similar plan to Amphioxus.
And in birds, like the fowl mentioned in the text, or mammals like the rabbit, they have even more yolk, or a history of yolk.
Right. In the fowl, with its massive yolk, gastrulation looks very different superficially. It happens in a small disc of cells sitting on top of the yolk, forming a structure called the primitive streak, which is thought to be equivalent to that blastocore region in the frog or Amphioxus. In the rabbit, which has lost most of its yolk secondarily, development still proceeds in a way that strongly resembles the fowl pattern, suggesting it inherited that developmental program from heavily yolked ancestors. The basic outcome forming the three germ layers, notochord and neural tube, is the same, even if the process looks different because of the yolk influence.
So the underlying plan is conserved, even if the details vary, and as development continues, you see even more fascinating resemblances between the groups.
Absolutely. Take the circulatory and respiratory systems, for instance. The frog tadpole starts life essentially as a fish. It has a simple, two-chambered heart pumping blood through aortic arches to gills for breathing underwater, a single circulation, just like the dogfish.
A fish stage.
Pretty much. But then, as it undergoes metamorphosis into an adult frog, the heart becomes three-chambered, the gills disappear, lungs develop, the skin becomes important for breathing, and the circulation changes to that intermediate double-loop system we talked about. Its individual development, its ontogeny, seems to echo its evolutionary history, its phylogeny.
That's amazing. And the embryos of land vertebrates like the fowl and rabbit have structures that fish and amphibian embryos don't.
Yes, two really key structures appear, the amnion and the allantois. These are extraembryonic membranes, meaning they're formed by the embryo but lie outside its main body.
What do they do?
The amnion grows around the embryo, enclosing it in a fluid-filled sac. This creates a private little pond, protecting the embryo from drying out and cushioning it. It's essential for animals developing in a shelled egg on land or inside a uterus. It's basically what breaks when a woman's water breaks.
Ah, okay. And the allantois?
The allantois starts as an outgrowth from the hindgut. In egg-laying animals like birds and reptiles, it grows large and spreads out just under the shell, acting as a surface for gas exchange, breathing through the porous shell, and storing waste products.
And in mammals like the rabbit?
In mammals like the rabbit, the embryo develops inside the mother's uterus and doesn't have a shell or massive yolk. It gets nutrients and oxygen directly from the mother. Here, the allantois plays a crucial role, but a different one. It grows out and fuses with the outer membrane, the chorion, and interlocks intimately with the wall of the mother's uterus. This combined structure, made of both embryonic and maternal tissues, is the placenta.
The placenta, that unique mammalian structure.
Exactly. It allows for incredibly efficient exchange of nutrients, gases, oxygen in, CO2 out, and waste products between the mother's blood and the embryo's blood, without the bloodstreams actually mixing directly. The text highlights the placenta as a really distinctive mammalian feature, pointing out that the egg-laying mammals, the platypus and echidna, are exceptions, lacking a true placenta. And it shows how this vital structure develops from membranes like the allantois, which are shared with other land vertebrates like birds and reptiles.
Okay, so putting all this together, these incredibly detailed anatomical comparisons showing shared structures, these really striking similarities in the way different groups develop from an egg, even the appearance of temporary structures in embryos that resemble features of other groups. What's the big conclusion that H.G. Wells draws from all this evidence in his textbook?
Well, he argues very forcefully that all this evidence, taken together, points overwhelmingly towards a single conclusion: common ancestry. He says it's not just that different animals were somehow created independently following a similar abstract design plan or adherence to type. No, the similarities, especially in development, are too deep, too quirky, too specific for that.
So rabbits, frogs, dogfish, they're related.
They're related. They aren't just built along a loose theme. They are, in his words, remote cousins, all descended through vast stretches of time from a much simpler shared progenitor organism. And based on the anatomy and the developmental stages, that primordial vertebrate ancestor was likely something much more like the simple Amphioxus or perhaps the dogfish than the more complex frog or rabbit.
Okay, common ancestry. But how does the change happen? How do you get from a simple ancestor to all these different forms?
The book then introduces the core concepts needed to explain that change. First, inheritance. The simple observation that offspring generally resemble their parents. Traits are passed down. Second, variation. The equally important observation that offspring are never exactly identical to their parents nor to each other. There are always slight differences.
Inheritance and variation, got it.
Now, these variations, these differences, can affect an individual's ability to survive and reproduce in its particular environment. Some variations might be harmful, some neutral, but some might be advantageous, making the individual slightly better at finding food, or avoiding predators, or attracting a mate, or resisting disease, whatever the challenges are.
So some variations are helpful for surviving and having kids.
Exactly. Individuals possessing these advantageous variations are, on average, more likely to survive long enough to reproduce, and to reproduce successfully, get on in life, as the text rather charmingly puts it. And because of inheritance, they will tend to pass those favorable variations on to their own offspring.
Okay, I see where this is going. This is natural selection.
Precisely. Over many, many generations, this process, the differential survival and reproduction of individuals based on their heritable variations, leads to what Wells calls a continual tendency for a species to become better adapted, more perfected for its specific conditions of life. And crucially, if those conditions change, if the climate shifts, or a new predator arrives, or a food source disappears, then the direction of selection will change too, and the form of the species itself will gradually change over time to adapt to the new reality.
Does he give an example to make this clearer?
He does, a really vivid one. Imagine, he says, deer being hunted mainly by tigers in a dense jungle versus deer being hunted mainly by wolves on open plains.
Okay, different environments, different predators.
Right, for the jungle deer hunted by ambush predators like tigers, maybe variations leading to better camouflage like stripes or spots, silent movement, and quick bursts of speed to escape that initial pounce would be favored by selection.
Makes sense. Stealth and sprint speed.
But now, change the conditions. Put the deer on open plains where they're hunted by wolves, who hunt in packs, often running their prey down over long distances using scent. Now, what traits are favored?
Probably endurance. Stronger legs for long-distance running, maybe bigger lungs. Stealth might be less important than just being able to outlast the wolves.
Exactly. Selection would now favor endurance, powerful limbs, a larger chest cavity for better respiration. Acuity of sight and hearing might still be useful, but perhaps less critical than sheer stamina. So the ideal deer form changes because the environmental pressures have changed. Rougher, bigger, more robust forms might now be favored.
So the species adapts to the new challenge.
Yes, and the text makes the direct link. This kind of transition from a tiger-adapted deer population to a wolf-adapted deer population happening over many generations driven by natural selection is exactly analogous in principle to the much grander transition from, say, a dogfish-like aquatic ancestor to a land-dwelling mammal like a rabbit. The only difference is the timescale. It's happening over vast epochs, millions and millions of years, instead of just maybe thousands of years. The mechanism, inheritance plus variation plus selection, is the same.
That's a powerful analogy. Does he bring in any other evidence?
He does. He briefly points to another completely independent line of evidence that strongly supports this whole story of gradual change and common ancestry. Paleontology, the study of fossils.
The fossil record.
Right. He notes that the fossil record, even as it was known then, shows a consistent pattern. The oldest rocks with vertebrate fossils contain fishes. Rocks from later periods show the first appearance of amphibians. Later still come the reptiles, and then, significantly, forms that look intermediate between reptile and mammals appear before you get the more modern types of mammals appearing highest up in the sequence.
So the order in the rocks matches the story.
It matches the story told by comparing the anatomy of living animals and by studying their embryonic development. Fish first, then amphibians, then reptiles, then mammals. It all fits together: anatomy, embryology, fossils pointing towards descent with modification from common ancestors over immense periods of time.
It really is quite remarkable, isn't it, how this textbook, written for biology students over a century ago, uses such detailed observation and careful comparison of just these few representative animal types of the rabbit, frog, dogfish, Amphioxus, to build such a clear, logical, and compelling case for evolution. It ties together anatomy, development, and even the fossil record.
It absolutely does. And it really reinforces Wells's argument, and the argument for the type system in general, doesn't it? That this kind of rigorous, focused, comparative study is perhaps the most valuable way to truly understand the big, broad biological principles like common ancestry and adaptation through natural selection. It's not just about listing facts. It teaches you how to look at life, how to see the connections.
This deep dive into H.G. Wells' biology text really shows us the power of focused observation and comparison, doesn't it? By looking so closely at just these few examples, the rabbit, the frog, the dogfish, and Amphioxus, and seeing not just how their systems differ, but more importantly, how they are fundamentally similar in underlying structure and developmental patterns, Wells guides his students and us today to grasp the profound connections between all vertebrates and the compelling evidence for life's grand unfolding story.
It really does. It takes what could be the seemingly dry study of anatomy and turns it into a detective story, uncovering clues about the history of life itself.
And it leaves you with a really powerful thought to ponder, drawn directly from the pages of this old textbook, something Wells clearly wanted his students to appreciate. Think about this. For a brief time in your own development, way back when you were just an embryo, you possess structures that strongly echo creatures like the dogfish things, like pharyngeal pouches, which are related to gill slits, or a notochord before your vertebrae fully formed. These are temporary features, vestiges perhaps, that hint at an ancient, shared aquatic past.
Echoes of our ancestors within our own development.
Exactly. It's as if the deep history of life is somehow condensed and partially rewritten in the story of each individual's journey from a single cell to a complex adult, a kind of silent testament written in our own bodies to those vast epochs of evolutionary change that Wells was describing.
That's a beautiful way to look at it, actually.
Thank you for joining us for this deep dive.